Myself Vikas Kauda. Warm welcome to our YouTube channel. Today we are discussing about another part of our lecture. Let's see about cell organelles. In last class we studied about fluid mosaic model, and in that we came to know what are all the aspects we learned about. Today let's learn about endoplasmic reticulum. Before going to endoplasmic reticulum, let's recognize the concepts of last class. I leave some concept which is, you people know the genome of mitochondria. What the genome of mitochondria is called? I didn't include in PPT. I will include genome of mitochondria is chondriome. And genome of plastid is plastrome. The genome of mitochondria is chondriome, genome of plastid is plastrome. By telling this, let's start about the cell organelle. Our first cell organelle is endoplasmic reticulum. The vesicles of 25 to 500 millimeter diameter is present in endoplasmic reticulum. In microtube, the tubules of 50 to 100 millimeter of diameter is present. You know tubules? Okay. In endoplasmic reticulum is not found in RBCs, egg, undifferentiated cell, prokaryotic cell. This is an important point for our exam point of view. They are all not present. They may ask in statement based question, endoplasmic reticulum is present in RBCs. Is statement 1 is true or false? Like that, statement wise questions can be asked in the examinations. There are many, there are two types of endoplasmic reticulum. First one is rough endoplasmic reticulum, smooth endoplasmic reticulum. Rough endoplasmic reticulum is outer surface is rough. Outer surface is rough and it is mainly composed of cystoma. Rough endoplasmic reticulum is outer layer of rough endoplasmic reticulum is rough and it is mainly composed of cystoma. And outer layer of endoplasmic reticulum, if it is smooth, then it is considered as smooth endoplasmic reticulum. Okay, let's check what is the functions of endoplasmic reticulum. Rough endoplasmic reticulum provides protein synthesis. It provides protein synthesis and it provides structural pair of lipids and phospholipid. And both rough endoplasmic and smooth endoplasmic reticulum transport the material to one organelle to another organelle in cell. It provides or control the passage or export of mRNA from nucleus. Export of mRNA means here translation or transduction concept comes here. It is application level. Translational level is controlled by which level? The organelle which can be studied in translation level or transcription level means it is endoplasmic reticulum. It is provide control over the transfer of mRNA to nucleus, from nucleus to rough endoplasmic reticulum. Okay, let's study in transcription translation process. Just remember the function. Now, the several enzymes are present in endoplasmic reticulum, such as glucose 6-phosphate ATPase. Here, ATPase also present. The main enzyme of endoplasmic reticulum is glucose 6-phosphate. Okay, let's move on to next organelle, that is ribosome. You people know it is a protein factory of cell. Ribosome consists RNA plus protein. Ribosome consists RNA plus protein. The synthesis associates with nucleolus. In nucleus, many chromatin network are there. In nucleus, nucleolus is present. The synthesis of nucleolus is by ribosome. The ribosome is attached to endoplasmic reticulum and also present in hyaloplasm, unattached to many membrane. Let's see about many groups of ribosome. Here there are three groups of ribosome. One is ATS in animal cell and another one ATS in plant cell, 70S in. Three groups of ribosome. One is ATS in plant cell, ATS in animal cell, and 70S in both prokaryotes and eukaryotes. Here S refers to unit called Schwetzberg unit. In animal cell, 80S subunits divides into 60S and 40S. In plant cell, same result, 60S and 40S. It subdivides into 60S and 40S. In prokaryotes and eukaryotes, 70S is divided into 50S and 30S. Here in eukaryotes, rRNA units are eukaryotes. rRNA unit is 23S, 5S, 56S. In prokaryotes, rRNA subunits are 23S, 5S, 56S. In prokaryotes, 28S, 18S, 5.8S, 5S. Just remember the subunits of rRNA of eukaryotes and prokaryotes. Move on to next topic, that is Golgi body. Golgi body is originate from endoplasmic reticulum elements. They are located near nucleus of the cell. Functions are protein transported to Golgi stores, modifies and packed, called packaging unit of cell. Here a ribosome is called as protein factory of the cell. Here Golgi body is called as packaging unit of cell because proteins are packed in Golgi store and modified in respective to cell organelles, and proinsulin is breaked to yield insulin. This is important point for examinations. How the insulin breakups, because this is important to exam point of view. Proinsulin is break down into functional insulin in Golgi body. Let's move to another cell organelle, that is lysosome. It is known as suicide bag of the cell. Here vesicles of 400 to 800 micron. 400 to 800 micron produced from Golgi. It contains several hydrolytic enzymes. The main enzyme of lysosome is acid phosphatase. This is present in lysosome. The primary lysosome produced by Golgi forms secondary lysosome and it is capable of digesting the living organism. That's why if any punctures in the cell organelles, it destroys itself. That's why it is called as the suicidal bag of the cell. Okay. Let's move on to next organelle, that is vacuole. Vacuole is present in both animal cell and plant cell. As we discussed, in animal cell it is small and multiple. In plant cell it is large and single. Here you know the cell membrane of vacuole is called as stonoblast. The matured vacuole occupies 90% of the cell in plant cell. 90% of cell. Okay. The matured and differentiated cell of vacuole occupies 90% of cell. It maintains it. What are all the functions of vacuole means? It maintains water pressure, support to plant and animal tissue. Let's move on next topic. Next organelle is centriole. It is cylindrical shape. As you people know, this is the characteristics difference of cell, animal cell and plant cell. This is only located in animal cell. It involves the spindle fiber organization or spindle apparatus. Here, without this, cell division is can't happen because spindle apparatus is very important in cell division. Therefore, centrioles plays the vital role in cell division. And next cell organelle. Here centrioles are. Okay. And come to next cell organelle, centriole. It is, as you people know, it is confined to only animal cell. This is the distinctive character of animal cell, and it is absent in plant cell. It involves in spindle fiber apparatus. It involves in organization of spindle apparatus, and therefore this play vital role in cell division. And come to next part, that is plastid. Here plastid, there are three plastids are there. One is leucoplast, chromoplast, chloroplast. Here leucoplasts are colorless and store of fat, starch and proteins are. These fat, starch and proteins are stored in leucoplast. And next, in plastid only, next type is chromoplast. Here in chromoplast there are two pigments are there. First one is phycoxanthin, which is yellow pigment. Second pigment is phycocyanin, which is blue pigment. And next part of plastid is chloroplast. This is very important, and you study all three because it is colorless. And what are all the components of chloroplast? What are all the pigments present in chromoplast? These are all the very important concept for your examination. And in another cell organelle called chloroplast, it is present in plant cell and it is double membrane bound organelle. Here outer layer of chloroplast is called as outer layer of chloroplast is called as thylakoid. And inner fluid like structure called as stroma. Inner fluid like structure called stroma, and these stacks are called thylakoid. One stack is called as thylakoid. Like that group of stack is called grana, and this play a very important role in photo reaction, that is dark reaction and light reaction, let's study in physiology classes. Here chloroplast have its own DNA, therefore it is called a semi-autonomous body, and it have its own ribosomal bodies. And outer layer is very permeable, highly permeable to organelles or liquid or nutrients. Outer membrane is highly permeable and inner layer is less permeable in plant cell. Let's move on to another organelle called mitochondria. It is called as powerhouse of the cell because, as you people know, mitochondria has its own DNA, which is also called as semi-autonomous body. It present in both plant cell and animal cell. The first term is coined by Benda. Some scientist names are also important in examinations. The term mitochondria is coined by Benda, and it is the autosomal body, semi-autosomal or autosomal body, because it has its own DNA and it has its own ribosome. Here in mitochondria, Krebs cycles occurs. Let's study Krebs cycle in physiology part. Next, your outer matrix is processed by here. This finger-like projection is called cristae. Inside this finger-like projection, matrix is called. Here many DNA and ribosomes are present. In matrix, DNA is divides as such as bacterial fusion, and ATP synthesis in this matrix as DNA ribosome is present. Therefore, it is called as powerhouse of the cell. Now let's move on to next cell organelle, cytoskeletons. Cytoskeletons are microfilaments and microtubules. It is present in both prokaryotes and eukaryotes. Microfilament means it involved in cell movement, and microtubules is composed of 13 fibers, which is made up of protein molecule, which constitutes the fiber and spindle apparatus, and these are all the main component of cell division, where the cell division occurs with the help of these organelles like chloroplast. Let's move on to another cell organelle, that is nucleus. It is very important concept for our examinations because it is the part where cell division occurs. And let's see the diagram. Here this one is nuclear envelope and the pore is nuclear pore, and this minute thing, this projection is called chromatin network, and this is nucleolus. Here nucleolus takes deep strain. Here nucleolus takes deep strains with basic dyes. It produces in nucleus. There are many ribosome, tRNA, mRNA. You know tRNA, transfer RNA, messenger RNA. The significance of nucleus is given by hammer link, and here cell composition is protein of 70%, DNA are small around, and from nucleus mRNA, tRNA, rRNA are produced. In rRNA, NOR is present. NOR means nuclear organized region. Let's study in transcription part of our class. And these are all the concepts which are studied under nucleus. Next, move on to next cell organelle. That is not cell organelle. It is comes under the nucleus, and it is chromosome. Let's study about chromosome. It is present in nucleus only. This constricted part of chromosome is called as satellite. These both arms are Armstrong or arms. This constricted region of chromosome is satellite. The both arms, this about chromosome. The constricted part is called satellite, and the middle part is centromere, which is used in the next cell division where spindle fibers are appears. This one is divided, and this is metacentric chromosome because centromere is located center of chromosome. Let's metacentric chromosome means centromere is located in middle of the chromosome where both the arms are equal length. Therefore, it is called as metacentric chromosome. Let's study about different types of chromosomes in cell division part. Thank you. If you want this PPT, check in description below and join to our Telegram channel. Thank you. Thank you.